Let's finish up our conversation about DNA replication by looking at proofreading and repair. It turns out that DNA polymerase can actually proofread the daughter strand while it's making it and replace any mismatched bases. So it has this opportunity, it's buzzing along at a really high rate. When it accidentally sticks the wrong nucleotide in the place, it has the opportunity to go back and actually cut that nucleotide out and insert the appropriate one. To give you an idea of how important this sort of proofreading and repair is, in just little E. coli's genome, there are at least 100 known DNA repair enzymes. So out of something like 5,000-ish enzymes, genes in all of E. coli, about 100 of those are code for DNA repair enzymes. Extremely important, and it makes sense from the perspective of survival. These bacteria and other living cells need to be able to faithfully replicate their genomes. In the human genome, it looks like so far we've found about 130 known DNA repair enzymes. With the proofreading that goes on with DNA polymerase, the error rate is about 1 in 100 million nucleotides. A wrong nucleotide will be put into place. If we knock out DNA polymerase's ability to proofread, that error rate goes up to about 1 in every 10,000. So basically what we're talking about is about a 10,000-fold decrease in errors and mutations in the genome during DNA replication simply due to proofreading. So this is a pretty remarkable phenomenon. If you go on to take microbiology, one of the things you'll learn is that RNA polymerases don't have any sort of proofreading activity. And what that means is that viral genomes that are RNA-based genomes, sort of like the influenza genome, actually are subject to a very high mutation rate, maybe something in the ballpark of 1 in every 100 million, or pardon me, 1 in every 10,000, right? Because they don't have the proofreading. And so what that means is that RNA genomes in viruses tend to mutate a lot faster, and how that plays out in a pathogen like influenza is that we have to get new vaccines every single year because there's this thing called antigenic drift as the genome drifts essentially during all these rounds of replication. So that's proofreading, extremely important component of DNA replication. Now, it turns out also there's an end-replication problem with these linear chromosomes. We've talked about how DNA primase sets down a primer and then an RNA-based primer, and then DNA polymerase picks up just a dozen or so nucleotides in and continues to extend that primer, but with DNA nucleotides. And then as DNA polymerase comes in from behind from another replication fork and bumps into the 5' end of that primer, it replaces those RNA nucleotides with DNA nucleotides, and then DNA ligase comes in and seals the nick. The problem is at the 3' end of a template strand, you will have a primer that will sit down and extend in its 5 to 3' direction. So if you look at this 1, 2, 3, fourth picture of DNA down, we've got a template with its 3' end to our right. The 5' end will match up. That RNA primer can be removed, but DNA polymerase can't fill in the gap because there's nothing to the right. There's nothing in the 5' direction to motor along and fill in that gap. And what ends up happening is that with each round of replication, linear chromosomes get a little bit shorter, maybe 10 or 12 nucleotides shorter. Think about that. If you've got slow-growing cells, it may not be a problem. If you have very fast-growing cells, such as, cheek cells or skin cells, round after round after round of mitosis is actually going to degrade and shorten the length of the new daughter strands, and the chromosomes themselves are going to be shortened. Think about that end replication problem. Maybe pause this video and make sure that makes sense to you. Go back to your textbook and kind of wrestle with the idea of why you end up with a gap of 10 or 12 nucleotides at the 5' end of each new daughter strand resulting in degradation of the ends of a linear chromosome. So the end replication problem has three solutions as far as we can see it. One would be to have circular DNA, like most prokaryotes have and like most plasmids have. You have circular DNA, there is no end, and therefore there is no end replication problem. There is always a way for a DNA polymerase to come in approaching the 5' end where an RNA had been removed and fill in that gap, letting DNA ligase seal the nick. So prokaryotes have a very effective conformation to their DNA. Eukaryotes have something called telomeres. Telomeres are essentially caps at either end. These are repeats of a 6-mer, meaning a pattern of 6 base pairs, and anywhere from 100 to 1,000 repeats at both ends of the chromosome. They don't code for anything, meaning you've created a buffer so that as replication, DNA replication proceeds round after round, and we edge away at those two ends of the molecule, we end up only eating up, hopefully, the telomeres, and we don't end up getting into any either regulatory regions or actually coding sequences of any genes. So telomeres are a very common and important solution among eukaryotes for dealing with this end replication problem that arises from linear chromosomes. And then one more component to this is that germline cells, these are stem cells that give rise to the gametes. In other words, they're the stem cells that are going to undergo meiosis and give rise to sperm and eggs. These cells have an enzyme called telomerase. And after each round of division, telomerase lengthens the telomeres, knowing that they're getting eaten away. And this is especially important in those germline cells, because if by the time a germline cell has given rise to some sperm after many, many rounds of division, it may be that those chromosomes have degraded. So this is a protein RNA complex that acts together as an enzyme. You might consider it a ribozyme, and its role is to make sure those telomeres themselves don't get degraded in the process of building our gametes. These are the three solutions to the end replication problem. There are some folks that have proposed that the shortening of chromosomes may be a major factor in aging. Take a look at James Watson on the far left in the 1950s, and then I found a picture of James Watson from 2007. Aging. Does shortening of chromosomes have anything to do with aging? There are some folks who believe that it does, and there's some pretty intensive aging research going on looking at telomeres, telomerase, and the shortening of chromosomes due to the end replication problem. So just something for you to think about. I hope you'll take the time to go through this video a couple of times, pause when you need to, take notes, go back to your textbook and fill in any gaps, and let me know if you have any questions.